Hey, Carlos.
Carlos, bom dia.
Hey, let's see if this works because I'm at the airport. So
Yes, I guess so. And you're flying during the panel discussion, right?
yeah, well,
Um,
let's see. Let's see if it's the only problem that I have is that it's this discussion. So that's why I asked Jacob if he could find someone else to do it. But in theory, I'll be able to be here around somehow.
Well, I can try to replace you during the panel discussion, but not during your presentation.
Okay, so we have some attendees already connected. I see the number is growing, so let's wait just a couple of minutes until everyone is here so we can
How
start.
many participants are we expecting?
We have 354 registered.
Good.
Let's see how many connect. Anyway, we are recording the session and we will make it available later on for those who cannot connect.
That's great because the people from GLOSOB requested if there was going to be a recording. So that's nice. Thanks.
So we have only 3 panel members. That's correct, Jacob?
Yes, that's right.
Okay. And because I'm now replacing Carlos, I was thinking, when we open the panel discussion, we give first each panel member an option to say something, right?
Yes. Maybe just a few minutes to introduce themselves, kind of where they link into soil biodiversity.
Okay, and then you are going to do the technical thing that if there's somebody who wants to say something, you can see that he or she will be, because I expect there's also some time for people, participants to say something or to ask something. Is that correct?
Yes. So if we'll ask them to put any questions that they have in there's a Q&A button down at the bottom.
Yes.
And then I will monitor that.
Okay.
And
perfect.
then as you're going through, I will pick some of those questions and I will forward those to you, so that
if you
don't have to monitor that.
If we can do it a little bit together, and then we can also emphasize that, of course, we can only address a few questions, but we keep the questions in the chat, correct? So if there are particular questions we don't have any time, we can respond later.
That's
either
right.
Panel members of ourselves, the board, so that they're not thinking, what a pity that it was not my turn, because the questions are stored. Yeah, and after the meeting, the board or panel members can have time to see their responses, something like that. Carlos, I see your hand.
Yeah, I just wanted to say that I'll finish my presentation with a vision of how this could work, and it's basically what we have discussed, Jacob, that we structure this from a global initiative to national initiative, where one contributes to the other somehow. And maybe just to give you a heads up, Peter, because if you are managing the panel, maybe this could be a way to start a discussion by exploring a bit or thinking about this idea.
Yeah, I think that I see that your connection is not 100% perfect, but we have to deal with that. It's already very nice that you can be with us from the airport. If there are any technical issues, your presentation, I guess, is also in Jacob's hand, so you can. Is that correct, Jacob? Do you have the presentation from Carlos?
I can send it. I can send it, but
if
I will try to
if you
improve
can
my
send
connection.
it, then if there's anything going wrong with the connection, we have your slides, we have your voice, so maybe correct, but not. And we have also Carlos, of course, as somebody who may contribute a little bit, or Jacob or whoever, so that we can manage your presentation quite well. And everybody will understand that when you're at the airport, things can go wrong. Yeah.
And it might go better. It might go better if you turn off your video so that there's not quite as much bandwidth.
Yes, Carlos.
Yeah, I will do that for sure.
Okay.
I will do that for sure.
And I already see some panel members joining in. Ederson and Adriana, very welcome.
Hi.
Thank you. Hello.
Very welcome. Nice of you that you want to join the panel.
Thanks. Thanks for the invitation.
Well, we are honored. And we also will, there's Ida. It gives a little minute to see whether I can pronounce your names well. It's Ida, correct? Yes? Okay, thank you, Ida. It's Adriana and Ederson, correct?
Yes, yes.
And if you start a panel meeting, you all three have a few minutes to say something about who you are, but also what you think about the things we try to develop, so people understand why you are on the panel. And so that will be part of the panel discussion. And thereafter, we will have discussions with the participants. And Jacob.
Yeah, so Yeda is not going to be on the panel.
You're not on the panel. Oh, yes.
Yeah, her information, George is going to present a lot of what she has done.
Okay.
Actually, Ederson, but that's okay.
Oh, Ederson. Yeah. Sorry about that.
Ederson will do that. Okay.
And there's one person, Clara, that hasn't got on yet, that is the third panelist.
Yes. Okay. That's correct. So the slide is correct now, right?
Yes.
The panel slide. Okay. No confusion. Well, I think we can start or not. It's time.
Yeah.
And you want me to start directly, Jacob, correct? And then I will pass the word to Natalia.
Yes, that's fine. I mean, the only thing I would need to do is just tell everybody that it's being recorded, but you can do that. And so
yes, do you that, and I know when to start. Yeah, because you're at the buttons.
Okay, Natalia, do you want to let me know when?
You can go ahead. We are already recording, so go ahead. We have 63 connected participants at the moment, but I think you can start.
Okay. Well, welcome everyone. Thank you for joining this webinar. We're very excited about some results and some ideas that we'd like to share. I just want to let everybody know that this is being recorded so that we can share it with participants that are unable to attend, as this is global and there are a lot of people where this is not the best time. And so I will turn the time now over to Peter de Ruyter, who's the chair of the International Network on Soil Biodiversity.
Yes, thank you very much, Jacob. First of all, everybody very welcome to this first webinar. Very nice you can make it. Jacob, can you put on my presentation? So, and for all of you all from all over the world, very good morning, good afternoon, good evening. Very happy that you can join this first webinar organized by the International Network on Soil Biodiversity. Can I have the next slide? And I guess that most of you will be familiar with what NETSOB is about and our objective, but just to summarize it quickly, of course, we try to strengthen the knowledge about soil biodiversity. It's important that we create critical mass for the implementation of the Global Soil Biodiversity Observatory, GLOSOB, that will come back in the program later. We want to make a contribution to the monitoring of soil biodiversity and the biological indicators we need to do so. And at the end, of course, we try to provide science-based decision-making processes at various policy levels regarding the soil biodiversity. Next slide, please. So we have now close to 1,000 participants in NETSOB, which is quite a few, and they are distributed over 4 working groups. And the first working group is how to measure soil biodiversity, and it is this working group who is in particularly organizing the webinar of today. We have a second working group, and that is involved with how to manage soil biodiversity. The third one is to economically value soil biodiversity, and the fourth working group is about the development of effective policy strategies. And the pie diagram at the left, that are the percentages of participants in the various working groups, and you can see that the first 2 working groups are the largest, both more than 40%. The other are somewhat smaller, and there are also quite a few participants who are not specially connected to one of the working groups. In our working groups, so they try to produce documents. Can I have the next slide? Thank you. The first working group is organizing a documentation of so-called SOPs, which are standardized operational procedures on soil health parameters. And that will come back in the program of today. We have a second working group focusing on how to manage soil biodiversity. And that working group is now in the process of creating a document with, on the one hand, some general, mostly scientifically based chapters on soil biodiversity management, but it also includes case studies from all over the world about how particular farmers or land users are now, as we speak, trying to manage soil biodiversity as best as they can. And working groups 3 and 4 are now closely working together to produce a sort of, let's say, white paper perspective review paper about ways to economically value biodiversity, and also what that can imply for policy-making processes. So we are quite busy in producing documentation, and a lot of the participants are already involved in these processes. Can I have the next slide? Just to show you a little bit about the organization of NETSOB. Of course, we have the participants. It's a network. So think about, there are thousands of people working together in this network, but we have just also for organizational matters, we have a board. And in the board, we have the chair, that's me, and we have the coordinator from the FAO, the Global Soil Partnership. And then we have a connection with the Intergovernmental Technical Panel on Soils, which is a scientific advisory board of the FAO, the Global Soil Partnership. We are strongly connected to the Global Soil Biodiversity Initiative and to the Convention of Biological Diversity. And in this slide, we also projected the faces of who we are. So you might know some of us, and you know, okay, these are participating in the board. So we have the coordination by Natalia and Jacob, and Natalia will also say something in this meeting after me. And then we have the first working group headed by George Brown and Carlos Guerra. The second working group by Zoe Lindo and Jeff Battigelli. The third working group, Gianluca Bagnara and Giulio Malocchio. And the fourth working group is chaired by Rosalina Gonzalez and Luca Montanarella. Yes, thank you. Can I have the next slide? Shortly about the program. So we are now opening it. So this is the first part, which is me. Then I will hand over the floor to Natalia, who will say something from the Global Soil Partnership Secretariat. And then we have the program, which is mostly about the work of Working Group 1. And George Brown is here, and he will give us an introduction and the background of all the work, and then he will go through particular aspects like how to measure soil microbial parameters, how to approach soil functional parameters, which involves particular soil processes, and he also will say something about the soil fauna. Then we go to the next session, and that will be about how to monitor, measure biodiversity on a global level. Jacob, our coordinator, will say something about the Global Soil Biodiversity Observatory. And then we go to the Global Soil Biodiversity Observational Network. Now, already in the names, you see these two activities are very closely connected, or are also working very strongly together. That will be done, presented by Carlos Guerra, one of the vice chairs of Working Group 1. And at the end of the program, we will have a panel discussion, and I will introduce the panel members when we start the panel discussion. Oh, it's here already. We have 3 panel members, which is Adriana Montanes from the Soil Microbiology Laboratory from Uruguay. We have Clara Patricia Vinica from the Amazonian Scientific Research Institute, Colombia, and we have Edison da Conchava Jesus from Embrapa in Brazil. These 3 panel members will come back at the end of the program, and that will give the opportunity to hear what they have to say, but also to have your questions and answers during the panel discussion. So, having said that, I wish you a very nice and interesting webinar. Again, I'm very happy that you joined in, and I will now pass the floor to Natalia. Please, Natalia.
Thank you very much, Peter, and welcome to all the participants and also the panelists and the members of the board. It's very nice to see you. It's a long time we don't see each other around the International Network on Soil Biodiversity, so thank you so much for joining. Well, what to say, biodiversity is key for the sustainable development and for the agenda that the United Nations has set for the next decade. And particularly soil biodiversity has a very big role to play. Many people speak about the importance of healthy soils. Even soil organic carbon has been quite high in the agenda, but without proper soil biodiversity, rich soils in organisms, all these cycles of even for soil organic carbon or nutrients will not be possible. So I'm very pleased that we are moving with this network. And today we will hear about all the work that has been done in the last 2 years regarding better understanding the status of soil biodiversity, what countries are doing for monitoring or for identifying soil organisms in their countries, and even some existing initiative and policies, recommendations, legislations, etc., to protect and sustainable manage soil biodiversity. As you may know, FAO and the Global Soil Partnership has been mandated by the United Nations Convention on Biological Diversity to implement the International Initiative for the Sustainable Use and Conservation of Soil Biodiversity, and part of that includes the development and implementation of the Global Soil Biodiversity Observatory. So I'm really happy that we are moving forward with these activities and that the working group on soil biodiversity assessment is working hard on advancing those activities. I think I have nothing else to say. Just listen carefully to the presentations and discussions. Thank you again for being here and participating in the webinar. Just remember that you have the Q&A box, so please feel free to put all your questions there and we will try to address them throughout the webinar. And you also have a chat if you want to interact with other attendees. Thank you so much, and back to you, Peter.
Yes, thank you very much, Natalia. It's also good that we are aware that FAO and the Global Soil Partnership in particular are just facilitating this network. Without the FAO, this would not have been possible. Thank you very much also for being here in your introduction. Then I would like to go to the first item of the agenda, session 1, and I give the floor to George Brown, who is the chair of Working Group 1.
Okay, thank you, Peter. Thank you, Natalia. Thank you, Jacob. Thank you for coming, all of you who are present today in this webinar. It's a pleasure to be sharing with you today some of the work we've been doing over the last couple of years almost. And it's great to have some interesting things to talk about today, which I hope that this panel discussion will raise. But first of all, we'd like to throw out some data, some information, so you can all have an idea about what's been going on. So I hope that you can see my screen now, and that is okay. Let me close this here so I can see what I'm doing. Okay, can you see the screen all right?
Yes, we can.
Great, okay. So I'd first of all like to acknowledge the many co-authors who helped work on this this survey, its development, its implementation, its analysis, and say that this is just part of what we did. We also had a big literature review, but since most of the results that we had in the literature review are quite similar to what we have here, this is what we're going to present today. So it's basically the result of a global survey, which the FAO conducted in March of last year. And 70,000 emails were sent out. We got almost 2,700 replies. The survey was quite long. It had 122 questions, not 123, but nobody answered all of them because they were geared to particular groups, particular aspects of these 11 sections of the survey. So there were sections on microbes, on fauna, on community level, on biodiversity inventory and monitoring, ecosystem services, different applications and threats to soil biodiversity, education and communication activities, and public policies and their knowledge by the different respondents. So we will only be addressing the first 8 of these briefly. So it was, I'm sure many of you received a survey and saw it. It was in SurveyMonkey. And the main objective really was to give an idea of the state of the art of the knowledge and of the work being done on soil biodiversity worldwide, identify who is doing what, where, and how, and then sort of what inventories and monitoring and mapping and databases were available, information on specific taxa and the functions of soil biodiversity, and reveal which methods were being used to study them. So this is for today, what basically we're going to be addressing, the part on ecosystem services or knowledges and education and public policies and stuff we are not going to address. So the survey respondents were from almost 140 countries, close to 1,350 institutions. Most of the respondents were from Europe, almost 40%. Around just over 20% from Asia, around 15 and 70% from 17% from Africa and Latin America, 12% from North America, not counting Mexico, and 4% from Oceania. So we had a few countries, just Italy, India, Brazil, and USA, who had over 100 respondents from those countries. The organizations that people were working for in terms of respondents were mainly educational. There were also a large number of research organizations, as well as people working for the government. Very few people were from NGOs or self-employed or from like private sector. Most of the people working at the local or national level, a lot fewer of those, were working at the regional or even the global level. Microbes dominated. They were the main focus of most of the respondents. So almost 70% of the respondents worked with microbes. Between 15 and 17, 18% of those people were working on micro, meso, macrofauna, very few on megafauna. Again, fewer on public policies and those aspects. There were quite a few people, again, working on functions of these organisms in the soil, on inventories and ecosystem services related to the role of these organisms in soils. Most of the people, again, were working in anthropogenic, so disturbed ecosystems, mainly annual crops, but also various other, pastoral or forestry plantations, etc., as well as in natural ecosystems, so mainly native vegetation. When we asked people about inventories, well, there were 50 countries with inventories, and these were mostly covering, again, microbes. So microbes rule in this regard, but there was quite a few quite a few of them also between 45, 46% working on micro, meso, or macrofauna, and a lot less on megafauna. And interestingly, a lot of people did not know whether there were inventories available for their countries. So there seems to be a niche there for communication of these data within those countries. In terms of monitoring programs, 48 countries had monitoring programs. This is the map showing the ones who had those. And again, most of these were at the national level, less at the local or state level. Again, microbes were the ones mostly looked at in these monitoring programs, and less in fauna, and of course, a lot less in megafauna. When we look at the particular groups of the microfauna, and I'm going to go straight on now into the second section, which is basically the results of the survey concerning the different groups. And I will let Ederson, who will immediately follow me, talk about the microbes and their functions. So I will speak here just basically about the fauna, and then we'll go right into the next part. So in terms of the microfauna, most of the people who replied were working with nematodes. A large proportion of them with free-living nematodes or plant parasitic nematodes, less with entomopathogenic or other nematodes, and a lot less with protozoa. Most of the methods used were, of course, funnels or centrifugation, flotations, decanting, sieving, etc. These Behrman, of course, was the most common, most used method, and these basically reflect the fact that most of the people who replied were working with nematodes. When we look at the mesofauna, we see, of course, again, the two main groups people were working at were mites and springtails, around almost 50%, between 40 and 50% of those. And then when we look at the following most common groups, there were ants and beetles, a lot less with the smaller organisms, enchytraeids or pseudoscorpions and myriapods. And when we look at the macrofauna, of course, the organisms that are visible to the naked eye, these were mostly with ecosystem taxa who are representing ecosystem engineers, so earthworms, ants, termites, and beetles. And then less with predators or detritivores like myriapods, of course, millipedes and centipedes, and predators, arachnids, and of course, a smaller proportion of the people working with plant feeders there at the end. When we look at the main methods for looking at mesofauna, we see that there were people using both. Most of the people were using both direct and indirect methods of collecting them and observing them. And in terms of the direct methods, these were mainly, surprisingly, hand sorting. Wet sieving was also quite common. And then we look at the indirect methods. Most of the people actually were using either Berlioz or Tolgren funnels versus more high gradient extractors like the Kempson apparatus, and also pitfalls, so pitfall traps, and less with Winkler extractors. When we look at the macrofauna, again, in this case, actually, direct methods were the most common means of collecting and observing these organisms. lot of people also use both direct and indirect, but direct were more common. And of course, hand sorting was the most prevalent method used for collecting macrofauna. And then when we looked at the indirect methods, in opposite to the mesofauna, it was pitfalls were more prevalent than, well, Burlese or Togren funnel extractors. And of course, again, high gradient extractors were less used, but winklers were more prevalent. And this has probably to do with a lot of the litter fauna. The same with the ones active on the surface, which are collected with pitfall traps. When we look at the megafauna, the methods were all over the place, but they were mostly observational. And the main groups looking at were rodents, and then of course ground-dwelling snakes and ground-dwelling birds, interestingly. So less with moles or with armadillos and that kind of stuff, or salamanders. And then we look at the methods used to evaluate the function, the roles of these organisms. Well, most of these people were actually using molecular methods to look at different ways of what these people are doing, what these organisms are doing in soils. Decomposition measurements were quite common. And then, of course, other less common activities like ecotoxicological tests or looking at bioturbation, for instance. When we look at the main use or why people were looking at what they were looking at in terms of what these organisms are doing in the soils, there were eight main functions, 8 main roles of these organisms that were targeted, and that's their importance as bioindicators, followed by nutrient cycling, their importance for plant growth, and then those aspects related to assessing biodiversity in general of these organisms, like inventories, composting, monitoring, inoculation, and then finally biological control. So to sum up, basically, that's it for my next to the last slide. Is of course this reflects what most people had already known in throughout the world, that most of the work done with microfauna is nematodes and protists, and the most common methods that are used for collecting those organisms. Mesofauna, mites, and springtails, but in this case also Microcoleoptera and ants appeared there. And these, of course, with Burlese, with pitfall and hand sorting, and the macrofauna with ecosystem engineers, predators and detritivores and plant feeders. And the main methods were hand sorting, pitfall, and Burlese funnels. So thank you for your attention, and I'll pass on the word right away to Ederson so he can continue with the microbes. And I will stop sharing my slide. Stop sharing. There we go. So thank you, and I'll go on. Feel free to share your slides, Ederson. Thanks.
Okay. Okay, just a minute. Okay, I need to allow Zoom to get access to my. Okay. Okay, now it works. Okay, can you see it?
Yes.
We can.
Yes, we can see.
Okay, so that
Yes.
works. Okay, thank you, everyone. Hello, everyone. Wherever you are. My name is Ederson. First, I would like to thank you for the invitation. George, Jacob, Peter, Natalia. My mission here is to show you some of the results of the survey that George mentioned, and going to talk about microbes, basically. So I'll be presenting some results of the survey regarding the groups that are studied and also their functions. Basically, you saw from the previous presentation that most of the respondents, they studied microbes. So microbes dominate this service. And as we know, they are responsible for several key functions in the soil, and also measurements of their activities are important to give information, to bring information about soil sustainability. And despite the large number of publications on this topic, we lack a comprehensive review about research groups, their functions, and so the aim of the work was to carry out this survey. And also we have results for a literature review, but I won't show them here because of the time we have available. But basically, when we talk about microbes, bacteria and fungi are the most studied taxa. This is not a surprise. First, due to their importance in the soil, historically they are the most studied groups. And also they have well-established molecular markers. And probably this is one of the reasons compared to fauna that you have more studies. Nowadays with molecular methods, you basically extract DNA, you sequence, and you can have information about your microbial community. So that makes things much easier than using traps and identifying bugs and so on. So this is the what we have about the taxa. And when we talk about methods, basically a little more than half of the respondents, they work with culture-dependent methods. Most of them work with culture and independent methods. I'm going to show that in the next slide. But basically, these results here, they show that people working with culture-dependent methods, they are studying genomics and the molecular biology of these organisms, and also carrying out phenotypic characterization. A few are carrying out studies with transcriptomics, metabolomics, proteomics, and so on. So basically, an explanation for that large use of genomic molecular techniques, it's the reduced sequencing costs that we have right now, and the increased capability of sequencing methods that have been made genomic sequencing available to many scientists. And the high use of phenotypic methods, this is basically traditional, and probably that can be explained by the polyphasic nature of microbial classification. So you need nowadays to carry out those methods and also associate them to molecular methods like genomic sequencing to describe new species and have a proper classification. Okay, now talking about culture-independent methods, 75% of the respondents using these methods, and high throughput sequencing, especially with Illumina technology, and metabar coding are the main methods used. And but despite that, what's interesting is that some people still use the traditional fingerprinting methods like the DGE in PLFA. PLFA actually works with phospholipids, so it's more complementary. But when we talk about DNA-based methods like DGE and TR flips, we still have people using those methods to study microbial diversity. The results here, they show a dominance of metabar coding and metagenomics, especially to study prokaryotes, bacteria, and archaea, with a dominance of 16S as the taxonomic marker and ITS for fungi. And also we have an expressive use of qPCR as a more precise method to quantify microbes. Regarding why people they carry out those evaluations. We have here basically some results, especially for studying impacts of management and land use for taxonomy and biodiversity surveys, and to study the impact of those organisms on soil properties and ecosystem services. We have other uses like use in biotechnology, pharmaceutical and food industry, pest and disease control. But the major uses are basically land use and manage impact, to assess land use and management impact, carry out taxonomics and biodiversity surveys, and to evaluate the impact of these organisms. And when we talk about applications, we had questions regarding that subject. We see that people work more with biological nitrogen fixation, phosphate solubilization, plant growth promotion, and also with bioremediation and the use of bioindicators of soil health. And also, the biodiversity inventories. So these are the major applications that people have been looking for. Okay, now regarding microbial activity and processes, and also microbial biomass, we see that when we talk about this subject, soil respiration and enzymatic activity, and the methods that most of the respondents they use, more than 50% of them they use soil respiration, enzymatic activities in their studies. We also see here nitrogen mineralization as a subject of study, decomposition methods, and also evaluations of mycorrhizal colonization. When we talk about microbial biomass, here we can see the methods that have been used with 46% of these people using the fumigation extraction method, followed by substrate induced respiration, and also fumigation incubation methods. When we go deeper inside some of these variables, when you talk about respiration, the methods that have been used are basal respiration and also field measurements with a dominance of basal respiration. For mycorrhizae, most of the people, they carry out evaluations of root infection. And when we talk about organic matter decomposition, the litter bag method is the most used. Now, when we go and look into the data about enzymatic activity, phosphatases and beta-glucosidase are the most studied here. What's interesting is that when you go to the literature survey, we have a different picture, but regarding the survey that we did here, phosphatases and beta-glucosidase are the enzymes that are mostly assessed here. And when
Ederson,
I'm ending. Okay,
okay, thank you.
Okay, yeah. And just to end here, we have the main users is basically what we saw for microbes management, taxonomy, biodiversity surveys, and impact of organisms, both for activities and biomass. And so as concluding remarks, basically what we can say is that scientists, they assess microbial diversity, mainly bacterial and fungal diversity, and activity to study management and land use impacts, carry out taxonomic surveys and evaluate the impact of organisms. A large proportion use culture-independent methods, especially metabarcoding and metagenomics, with roughly half of them still working with culture-dependent methods. Microbes are basically used for plant growth promotion, especially biological nitrifixation, and as bioindicators of soil health. And regarding activities, basically soil respiration and enzymatic activities, they stand out as the most used methods. Okay, that's all. Thank you very much for the opportunity.
Thank you very much, George. Thank you very much, Ederson, for presenting this. There might be some questions, but we have the question session during the panel discussion. And again, this webinar is recorded as well as the presentations. The questions are stored. That means that if we run out of time to address all the questions, we have them, and it's also possible to respond to these questions later after the webinar. So for now, I would like to continue with the next item on the agenda, and that will be a presentation by our coordinator, John Parnell, about GLOSOB, our Global Soil Observatory. Jacob, please.
Yeah, Peter, I just got a message from Carlos asking if we could swap so that he could give his first, as he's got a time crunch.
Okay. Yeah, then also for you, Carlos is at the airport because of changing in flight schedule. So we are very happy that we have still him here giving his presentation from the airport. So we swap and we give first the floor to Carlos and then we go back to Jacob. Carlos, can you hear us? Then you can start your presentation. And Carlos, you're still muted.
Sorry, I just needed a second to set myself up. Here you go. So, my name is Carlos Guerra. I hope that you can hear me well and without all the noise that I'm hearing on my side. The idea of today is to show you a bit of how we are working in SoilBON, also in partnership with NETSOB, to develop what we think is a necessary global scheme to monitor soils that also supports countries in their monitoring of soils and soil biodiversity specifically. So what I would like to start is by saying that although we are planning this as a global initiative, actually what we are trying to aim is for an international approach that can actually foster local action. That's the point. And the reason for this is that we strongly believe that we cannot do the national monitoring that we aim to if we don't consider our position in what is happening in our neighboring countries. This requires standardized procedures for both sampling and analytics. This also requires establishing international cooperation in the scope of Nagoya, so to make this legal and to protect the interests of countries that are highly biodiverse. This also has a direct impact in the economies also, because we are talking about soils. And soil, contrary to air or water, is not a public good. Soil is actually a private property in many countries in the world, if not all. And there's a large proportion of the territory also that is private property and it's privately owned. So when we are talking about monitoring soils and what we can do to soils, we also have to think about this potential impact on humans. And this is the point that I will leave here, because soils are also made of people and of culture. In many places in the world, soils plays a very important role in the cultural background of the local people and the local society. So, because we are focusing on standardization and how to approach this at this higher level, I would like to start by trying to clarify what essential biodiversity variables are. They are not the thing. This is very important. It's very important to say, because these essential biodiversity variables only represent what we consider to be the minimum set of variables that we need to describe, in this case, the ecology of soils. And this minimum is important because this means that we have to cover different aspects of the ecology of soils, but this is not the limit. So we can do way more if we want to and if we need to, but at least these we should do. That's the idea behind this. And the concept actually stems from an activity from GEO that is called GLOSOB, and these Global Biodiversity Observation Networks already structured how these essential variables should look like into a few classes, and then they gave already some examples for this, let's say for terrestrial and non-terrestrial systems, but in a generic way. What we tried to do in SoilBON was to focus on the soil realm and develop what we describe as soil essential biodiversity variables. These include, to your left, variables related to function, because we need to characterize the function of soils, but also include, to the right, variables related to diversity and to the habitat of the communities that we are studying. The idea behind this is to create a system that collects information on different aspects of soil ecology, introduces them or allows for the calculation of indicators, ecological indicators, that we can then use to make a change in our world. That's the idea behind these essential variables, that they are at least the bare minimum that we can use to promote the sustainability of the ecology of soils. And this is independent of land use type. So the idea is that we can do this in agricultural soils, but also in forest soils, in urban soils, and so on. At SoilBON, what we aim is for full standardization. So this means that we have one global activity where the soils are actually transported to a central lab, and from this central lab, the samples are then divided and split into multiple other labs that do one single analysis or a few analyses on all samples. The idea behind this is to promote full standardization of all the steps, and this includes at the beginning of this, standardizing also sampling. So we have a very, very simple and very, very easy to use sampling protocol. And the idea behind this is to put at the same level everyone in the world. It doesn't matter if you have the financial resources of Germany, or if you have other limitations in your specific context. The idea is to provide a method that can be used everywhere. Not only a method that can be used everywhere, and if you are a SoilBON member, you already received one of these boxes. The idea is that we actually provide you with everything that you need to do the sampling. Not only the sampling scheme or the sampling method, but also the materials, even rubber gloves and a knife if you want, if you don't have a means to excavate soils, for example. The idea is to really level everyone, put everyone at the same level, and to allow for this scheme to be as simple as possible to allow for this standardization effort. But standardizing also means methods. And here, the presentations from George and his colleague before are really important, because at SoilBON, as I said, we standardize these methods at this global scale. And we don't go for the best available gold standard method, also because it's not cost effective in many cases. We go for the methods that we realize that can be, in some cases, more simple to implement across different geographies, but also we go for methods that we have available in a cost-effective way in the different labs that collaborate with us. This doesn't mean that everyone should do exactly as we are doing here. And in many countries, some of these methods will not be possible to implement. And in some other countries, the methods can even be better, or you can even do some analysis on other things that you think are for example, other taxonomic groups, other functions that you think are even better to represent your soils. The idea behind this is to have a standard at a global scale that we can then build on by having the national systems contributing to this global effort by providing also samples. And by doing that, we can then create transfer functions from what we are trying to observe at the global scale to what we can observe at the national scale. This would allow us to also to merge national systems into one big pool of data that we can then better understand what is happening in the soil. So this is the vision that I would like to more or less leave here with you, is that SoilBON, in collaboration with other initiatives, is trying to create a global scheme that should not function alone, because we will never be able to tell what is happening in your specific country. Sometimes, in some cases, for your specific region, maybe, but even though the sampling effort that we require is really low when we are talking about individual countries, we are trying to describe global patterns. We don't aim to describe national patterns. For that, we need countries to develop their own systems, and ideally in line with what SoilBON is doing, to create this matryoshka, if you want, of systems that can generate information at the national scale, but also upscale it to the international level. But for this, we need scale integrated soil monitoring systems. So this means that a national system communicates with this global system by sharing samples and by sharing data. And sharing samples here is important because this global activity should remain independent and research driven. The idea behind this is to provide the fundamental trust on how this system is operated and on the results that we are obtaining. The other aspect here is that we have to invest strongly in capacity building and in generating operational resources for everyone involved here. Countries will need to implement their systems, and in many cases, they lack the capacity or the financial capacity or the expertise to implement this at some specific level. We have to facilitate ways to overcome this. One way to do this is to also have the help of countries to facilitate international monitoring operations. This means, for example, overcoming several of the current Nagoya burdens that also apply to soils and that limit to some extent our capacity. Just for reference, in the last 3 years, since we started implementing SoilBON, the person who I talk the most are not necessarily ecologists, but lawyers. Lawyers that deal with your own institutional problems, with my institutional problems, and with international law. And that's not the reason why we created SoilBON. We created SoilBON to overcome some of these issues. And then lastly, we have to promote local experts in a network fashion, because in your country, maybe you don't have a specialist on earthworms, but maybe your neighboring country that speaks a close enough language has. And if that's the case, we have to connect these people. We have to allow for this cross-boundary type of research and transfer of expertise. Just to as my final slide, I would like just to show you that SoilBON is a network of people, mostly and foremost. We are all soil ecologists, all soil enthusiasts that work together and collaborate to promote this global network. We don't fill all the gaps in the world, for sure. There are many places in the world that are facing wars, are facing other dangers that make soil research even not a priority at all. But when we can and when we are able to support local researchers, we do. And this is important for us because all our data is open source. Everything is open data. And we, to the point that even our protocols, our images, our figures, everything is open to the community so they can develop their own systems in their own countries. And that's it. Thank you very much.
Well, thank you very much, Carlos, for your presentation. It's always impressive to see such projects which are not only very open to everybody, but so inclusive by removing all kinds of technicals or financial constraints so that we can really reach out to everyone who likes to be involved, that it is possible for them to be involved. Very nice presentation. Thank you very much. I guess you have to run to the gate to catch your flight. So thank you very much to be with us from the airport. And there were already some questions to you. I say it also to those who have posed these questions. They are stored in the chat. So Carlos can catch his flight and the questions can pass to him after the meeting. And you are likely to get the response after this meeting. So don't worry about your questions. The response can be made when this meeting is done. Again, also, George, thank you very much. You will be with us during the rest of the seminar, also maybe by answering questions that come up during the panel meeting. So then we go to the next item on the agenda, and that's about GLOSOB, and I will pass the word to our coordinator, Jacob Parnell. Jacob. Jacob, you might be still muted.
All right, you see my screen now?
But now we lost your screen.
Oh.
But we hear your voice.
Let me see if I can.
All we see is a dark background, nothing in the front.
Let me try this again.
We did see it.
You did see it, and then it stopped.
Yeah, when you unmuted yourself, then the screen was gone.
Do you want me to share it, Jacob?
Let me try this really quick and see if I can.
Yes.
There we go.
There
Great.
we go.
Okay. So thank you. Sorry, what was that?
It's not in full mode though.
We see background information there. Just try to make it.
We see the chat. Yeah.
You can increase the size so it fills the whole screen.
Okay, I'm not sure what's happening because from mine it looks like it's full screen. Let me try one more time before I bring Natalia in to rescue me.
Well, you know, we could see it. It was not perfect, but it might work. So this is
there we go.
Now it's perfect. Yeah.
Okay. Now that you've got full screen.
Yes, now it's perfect.
Okay, thank you, and I apologize for the little bit of a technical delay, and maybe it was a good thing that Carlos went first, because I want to introduce something that the FAO has been tasked with. So back in last December at the Convention on Biological Diversity, the COP15, 190 some odd countries signed on to an accord basically saying we need to protect soil biodiversity. And we need some way to be able to monitor it, to be able to evaluate soil biodiversity. And all of those countries signed on to that. And they signed on to charter the Food and Agriculture Organization with organizing a Global Soil Biodiversity Observatory. So the goal of this is basically to monitor and forecast the conditions of global soil biodiversity and how our human activities are impacting soil biodiversity and its role to function in a capacity of human health, of environmental quality. And so this is very much sounding a lot like what Carlos just described as SoilBON. So some of the tasks that we have are first we need to standardize the measurements. We need to figure out what measurements should we be collecting, and so George and Ederson's inputs on this were and are extremely valuable in helping us to figure out what are the most important things that we should be measuring. What are those essential biological variables that we should be measuring globally? And so we need to measure these, and this, of course, needs to be country driven. So this is going to be taken back to each one of those countries that signed on to this Convention on Biological Diversity to be able to say, here is the program that the International Network on Soil Biodiversity with the FAO is recommending that each country implement. And so part of this will be to bring out these measurements, but another part of it will be to build the capacities. We recognize that not all countries currently have the capacity to be able to measure everything. And so thankfully, the Global Soil Partnership has other networks, other programs that are there to build capacity. And so we will work with these other groups, these networks that have already been established, that are already working, that are already reaching thousands of different individuals to be able to start to build that capacity to be able to make the measurements that we are collecting. So this is kind of the flow of the Global Soil Biodiversity Observatory. And so the first thing that we need to do is we need to define these essential biodiversity measurements. And right now, what we are proposing to do is we are proposing to use those measurements that Carlos just mentioned, those SoilBON measurements that have already been adopted, that have already gone through the review, that have already been filtered through years of discussions and debate. And then we would have 3 different levels of membership to this Global Soil Biodiversity Observatory. And each one of these is designed to build up to that next level, eventually ending up in this full membership. And that full membership is that you are measuring all of those essential biological variables that Carlos mentioned. And so we recognize that not everyone can do it now. And so they would sign on with a subset of those variables. And GLOSOB, the International Network on Soil Biodiversity, NETSOB, different organizations that FAO has will work with those countries to build the capacity, to help train technicians, to bring in the infrastructure that is needed to be able to work their way up to moderate and then to full membership where they're measuring all of those variables. And then as they measure all of those variables, as Carlos mentioned, all of those would be able to feed into kind of a larger a data set so that we have a global assessment of biodiversity at the end of this. And so you can see that one area is to work with the countries to determine that membership. What biological variables are you capable of measuring now, and what do we need to work on? What do we need to help to build that capacity? And we would then build that capacity. We would increase the capacity until they are at that full membership level. And then we would have these national monitoring programs within each one of these countries that is standardized, that is doing the same measurements, the same SOPs, just like SoilBON has basically set kind of this standard, set the template for what we hope to do on a countrywide basis. And And so this is kind of the pattern. And so what I would want to do right now is just spend a few minutes and talk about these essential biodiversity measurements that we propose to collect.
Can you be quick then, Jacob, because otherwise we may run out of time.
Yep, I've just got I think one slide after this. So basically we're going to adopt this is the paper that Carlos was talking about that outlines those essential biological variables. So these are those biodiversity measurements that we are proposing to collect through GLOSOB. And so these have already been vetted through SoilBON. These have already, and SoilBON has adopted a lot of these from projects like the Earth Microbiome Project. And so a lot of these are kind of the gold standards that have already been set for quite a while. I've highlighted in black several of these that are already FAO standards. And so we have a Global Soil Laboratory Network where these laboratories have received these SOPs. They have quality checks that they go through. And so I think there are over 1,000 laboratories across the world that are standardized to these measurements. So soil respiration, bulk density, pH, CEC, where we would be working with them to give them some of these SOPs so that then they could start giving these SOPs out to these laboratories so that they can start building the capacity in those laboratories to start measuring these at a national basis so that we can have those national monitoring programs that feed into this Global Soil Biodiversity Observatory. And I think the last slide is just kind of so I'm just going to leave it on that just so that if anybody needs to write any of these down or look at any of these, they're available. But
Thank you very much, Jacob.
You bet.
It's very interesting to see how, on the one hand, an initiative like SoilBON is taken up by the FAO Global Soil Partnership by creating such a facility, GLOSOB, which will surely enhance and make SoilBON kinds of activities more successful. Both of them show that soil biodiversity monitoring on a global scale has gained a huge momentum at this time while we speak. And I hope that these two presentations also will encourage participants also of this webinar to join in. Thank you very much, Jacob. There are some people raising their hands. We cannot manage that. I'm sorry, there are too many participating members in this webinar, but you can put your question on the chat and they might come back during the panel discussion or maybe afterwards the webinar. So don't raise your hand, but use the chat or question and answer option. Then we go to the last item on the agenda, which is the panel discussion. And Jacob, if you can present that particular slide again, just for a short moment, then I can introduce the panel members to the participants, please. I did it already at the start of the webinar, but because some people have joined later in. Jacob, do you have that slide?
Yeah, I'm trying to bring it up now.
Perfect. Yes, thank you, Jacob. So we have a panel discussion and we have 3 panel members, which is Adriana Montanes from the Soil Microbiology Lab, Montevideo, Uruguay. We have Clara Patricia Venega from the Amazonian Scientific Research Institute, Sinchi, Colombia. And we have one of the speakers of this webinar, Ederson from Brapa in Brazil. And what we want to do in the last item on the agenda, that is that first we give the floor to the three panel members, that they have a few minutes to say what they like to say about themselves, but also how they look at initiatives like SoilBON, GLOSOB, and all the monitoring activities like the survey. And then in the remaining of the time, we have some questions and answers as much as we can. So first, Adriana, very welcome here. And
hi, everybody. Hello, thank you very much for the invitations. I'm very glad to participate and hear all this news and work that you are doing and the effort on the studies of soil biodiversity and status in the world. I'm in Uruguay. I work in the laboratory of soil microbiology. Well, what to say about that has not been said. First of all, I think I'm part of the SoilBON initiative, and when this initiative started, several scientists in my country put their hand up. And even if we are a small country and we know each other, the collaboration within institutions and maybe different disciplines is not quite easy. I think this is a quite challenge everywhere. But when this initiative started, we realized that we're several scientists working in the same country, and we took this chance to catalyze collaboration within the country. So actually we are 5 scientists working in the SoilBON initiative, and we create our own project with the science. We call this project SoilBON Plus. And because Uruguay is one of the we are on the Rio de la Plata grassland regions. That is one of the largest area of temperate, humid, and subhumid grasslands biome in South America, and one of the largest in the world. We think the results and the information regarding what happened on soil microbial diversity below the Pampa soil is quite important for us and quite important for the world too. And the integration of our results within this global framework will give us benefits in terms of how to understand what is going on locally and globally. Because in particular, I think is very important in soils, because the effect of soil degradation and the resulting environment impacts go beyond national borders. And this is a global problem. And a regional approach or a global approach will be best suited to understand and manage degradation process and develop effective conservation and restoration strategies. So our knowledge on soil microbial diversity below the Pampa soil is limited, and I think this to work together is quite important. We have this project, that we designed in our country. We follow the same methodology that SoilBON. We include another variables, and we include more sites. So we can cover 7 ecoregions in the country. And we cover protected area and degraded area. Grasslands has historically been degraded by intensive agriculture, forest, commercial forests. So it's important to have both situations and have the opportunity to recover the information on not only soil biodiversity. In our case, we do soil microbial diversity. We also measure functions, which are quite important to understand the soil health. So functions we measure related to soil nutrient cycling, like mineralization, soil enzymes, and all the variables that were measured here. I think it's quite important. So the challenge that we have, I mean, trying to sum up where we are, is to work the interdisciplinary and interinstitutional work is something that we have to cross lands and cross borders in terms of knowledge, especially when we talk about physical or chemistry of soil, which is quite important because it's the habitat of soil life. So we have to understand this aspect, and then we need another scientist working in these other disciplines. Not only more specialists on soil biodiversity, which is quite important too, but also on specialists that work in the soil habitat, which is the physical of soil is very, very important for the soil life. Other challenge, sorry?
No, go ahead, but I want to save some time for the others. So please.
Another thing is...
we have still 50 minutes.
Okay, is to develop indicators that maybe are not only scientifically sound, but also quite easy to apply for practitioners, or maybe we can seduce policymakers to be using monitoring programs to monitor soil biodiversity and soil health in the countries. Our project tried to develop these indicators, but also I think it's important for policymakers and society to understand or have or put on the table of the policymakers the problem that we have on soil biodiversity loss is the studies on the economic of soil health. I think is a working group on that. I think it's a small group, but the result of that group, I think, will be quite challenging to support the work in local work and international work on soil biodiversity. And well, I think that in terms of dissemination of knowledge, so how we create this bridge between academic knowledge or what we're doing in research with the really practitioner is quite important to create this mechanism. In our country now we are trying, I mean, we convocate a meeting. Also the ministries are quite interested in our work, so we are trying to do this, to generate this bridge, to generate these interactions. And
okay, Adriana, I'm sorry, but I have to
thank you.
A little bit short, but I thank you very much for your contribution. Very encouraging, and to hear what you're doing also from Uruguay, but also for the rest of the world. I want to pass the word to Clara. Clara, please. Is she here? Jacob? Is Clara here?
She has two tabs present here, so she should be connected.
I am here.
There's Clara.
Hello.
Hello, Clara.
Hello, I'm here. Could you hear me?
Yes, we can hear you. You can start.
Okay, hi everybody. Yes, thank you so much for this short invitation. Well, actually, I want to say is that yes, we started to study the biodiversity of soils, specifically of Amazon soils, here about 10 years ago. And what we discovered with the time is that we had a lot of information to share, but we were isolated. One of the things that opened and opened us to contribute in an international way was the SoilBON project. So we are there, and for us, it's very important the logic and to process information in the quality to understand more the soils, the biodiversity of soils, specifically the microbiology of soils. But we also discovered that we have a lot of information about macrofauna, soil macrofauna, and we are contributing with two other projects, the project Soil Fauna and the project Fauna Services. Those are international projects. The first is a global project, and the second is for neotropical ecosystems. And what we, the advantage that we have to participate in those international initiatives is that we have standardized global methodologies that help us to compare our information with information of other international institute and international laboratories. So I think that is very, very important. We think that we need to communicate more among ourselves to improve what we already know about the soil biodiversity. And I think we can do that. For example, in the case of mycorrhizal fungi, we have a very interesting network that goes more beyond the borders of our countries. That is the Mycorrhizal Research Network for South America. So in this case, it's a very spontaneous network in which we are really contributing a lot in the understanding of this specific symbiosis, microbiological symbiosis. So I think that's the root. And these spaces are very important for us because help us to show our results, our information, and also to analyze in a better way the information we have and put it in a global context that I think is the most important thing. That's only one I want to say. And thank you again for this space.
Thank you very much, Clara. The third member of our panel is Ederson. We have heard him before. Ederson, do you have something to contribute before we go to the questions and answers, please?
Okay, good. How many time do I have? It's about 5 minutes.
It's just a few. We have time. We have only 7 minutes, so it's nice that we can handle, let's say, two questions before we have to close.
Okay, that's more than enough. Okay, so there was a question, a written question about the challenge of standardization and bringing things together. I think that's an important issue to tackle, and actually that's the objective of Global Biodiversity Observatory. We saw some initiatives like SoilBON. We also have NETSOB. We have national initiatives, but something that I think that it's important to take into account. It's the different realities that we have in our different countries. For some of our countries, it's easier to carry out some of these evaluations, but it's not that reality for everyone, even inside a country. My country, for example, Brazil, we have people with different capabilities. So it's not easy to determine some methods and to tell people, okay, you should use this method. So I see these initiatives like SoilBON and NETSOB as important, although they won't solve this problem. We have also some examples in Brazil. One, it's Ieda is coordinating. She and her group and several other colleagues, they are working with a network to measure soil health. So they develop methods, standardized methods to make sampling easy for farmers and also for labs around the country to carry out this analysis. So we have some examples of, I think that we need to share these experiences but taking into account these realities. So and also keeping independency. That's what I think. It's good. Sometimes centralizing is good because it's easy to manage things, but it's important that each country and researchers, they can have independency about their own data and how they give access to that. And so I think it's a challenge to think about a way where we can be more collaborative. Okay. So that's what I would like to say. Okay.
Thank you very much. Thank you very much. So we have a few minutes. Again, don't raise your hands. That's frustrating because we cannot handle them. You just post your question on the chat. They will be stored and can be answered after the webinar. Carlos is already busy with that. Jacob is also selecting some questions, but let me then start with the first question that came in, and maybe that was a question for Carlos, but maybe also other people can respond to that because it has a general importance. That's the question of Alberto Acedo, and he asked how many samples do we need to represent a particular surface area? I think that's a practical problem many of us are facing. Is there anyone who is willing to respond to that? And I'm also looking at George.
Samples, how many samples? You can go and answer, George.
I read that question. It's
because
a big
Carlos
challenge.
Is gone and you maybe in that.
Yeah, I think in the sense that if we want to assess variability, we obviously have to have a lot more samples. But what has been proposed in SoilBON are composite samples over an area that goes basically, I believe, 30 by 30 square meters. So it's representing something that's basically a small plot. This is being replicated, of course, across different areas in the country and across the world. And I think the reason is to sample a lot of parameters within the same area and associate that with the vegetation. In this case, it's all native vegetation. So the question of how many samples is always a touchy one. And I think that they have addressed this in a sense by doing composite samples and at least standardizing how they're being sampled across the world.
Okay. Ederson, you want to say something additional to this? Otherwise, we have another question.
No, that's basically, it's not regarding SoilBON, but what it's recommended is to have pilot studies so we can define our sampling effort. We know that's not the reality for everyone, and most of the people don't do it actually. But that's simple. The answer is simple. You should adapt that to your reality. Go sampling, do small sampling, check if you are able to detect your biodiversity reasonably with rarefaction analysis and things like that. But that's not what people are doing. Okay.
I see in the chat that answers and questions are popping up. There's another question selected by Jacob, and that question is how can we assure the quality assurance and checks between labs? Anyone of the panel who likes to respond to that? So how can we assure the quality of the measurements and check that among labs?
Maybe I can say something.
Yes, is it Clara?
Well, yeah, well, you have to have, well, standardized methodologies, but also some indicators of the quality of your lab in the sense of that the equipments you are using are the correct, that the reactives and whatever you are using are correct, are working well, and that also that the professionals are using the methodologies and doing all the process are qualified. I think that would be great, and there are also some issue ISO no ISO I the word in English, but that also including have some quality standards that are qualified also for international organizations that say that actually what you are doing is what it has to be doing in that process. That's what I will say.
Okay, thank you very much. So we are coming to the end of the meeting. So I want to close by giving Adriana a short last word regarding this question, because I noticed she wanted to say something. And then because I see the participants are already leaving a little bit, then I want to close this webinar. Adriana, for you.
No, just wanted to add something to what Clara said, that what we do here is when we send samples to different labs, so you can check if you are working well. So it's like we call interlab. So the same sample go to different labs that doing the same methodology, and we check if the standard deviation of our results is more or less a bit good.
Okay, thank you. Well, I want to thank you, of course, being part of the panel, Clara, Adriana, Edison, but also, of course, because of all the work you're doing for soil biodiversity. I also want to express my thanks to the other participants in presentators in this webinar, George, Carlos, Jacob, and Natalia, of course, and also thank the Global Soil Partnership and FAO for making this all possible. Again, I hope you will get response to the questions which we can study afterwards this webinar. We had more than 100 participants in this webinar, which I think is a success. Thank you very much. More seminars or webinars will follow from the NETSOB. I would say it's a network. Let's keep in touch. And thank you all for being with us. Have a nice day.